I'm waiting for everybody else to join. I'm waiting for everybody else to join. I'm waiting. So let's see. What I was told was not every one of you are able to get to the stream chat. Because of that, um, hmm, I don't know why it's not going live there. But yeah, so I think you guys have to upgrade your operating system or your maybe Chrome or whichever browser you're using. You need to upgrade that. Okay, so I don't know. My video is not moving on the live screen. I don't know why, but um, I'm hoping you guys are able to hear me. Okay, so for some reason the video is stuck. Yeah, that's weird. Hmm. I don't know. My video got stuck here. Hmm. I don't know. The other technical difficulties that I'm having this morning is the screen that I usually scribble with a pen and circle and everything. That's not working. Okay, so I guess we're going to have to put up with that today. And the reason why I won't have quiz today is because not all of you are able to join through stream chat. So by upgrading your system, I know. Oh, it's not stuck for you. Okay, so I thought on my screen it's stuck. So maybe you're okay there. Okay, so the so tomorrow morning we'll have quiz. So can someone who was not able to join through stream chat, but are joining through today you are able to join through stream chat? Can you say that I was not able to, and I need to figure out why you're not able to do via stream chat. So people, oh, Vishnu is saying that maybe Firefox, people who are using Firefox, they have a problem. Is that true? Can you guys use Chrome and get to the stream chat? So yeah, so Afia, can you tell me how many people are still not able to get to stream chat, and they have to join through both, because I don't even check boardroom because it won't show up on the screen unless I go and click on it. And I think everyone should be able to join via stream chat one way or the other. Okay, so okay, for some people Firefox works better than Chrome. As long as you guys are able to join, so text me on WhatsApp if you are not able to join. Continue to not able to join through stream chat. Okay, we're going to have to do something about it. Okay, so tomorrow we are having quiz. You will not be able to take the quiz if you're not able to join through stream chat. Okay, so we'll have 20 questions. It is a multiple choice. So for each question, you will get 20 seconds to answer. And the moment you answer, it will score it, and it will give, by the end of the quiz, you will have a score. Okay. So if you didn't answer, it won't score you. Okay. So that's how it is. So again, I told you already that the other difficulty I'm having is with my PowerPoint slide. So I'm going to go into my PowerPoint mode right now. Okay. So the only thing I won't be able to do today is scribble with my pen, as I always do, which I really like doing, but I won't be able to do that today. So, but other than that, around 8 people are unable to access Stream Chat, ma'am. Few are using the personal computer as they're able to. Okay, okay. So we'll have to see how we help them to do it. Maybe. We'll figure that out. Okay, let's start the lecture. So today we are going to talk about all um, annotation aspect of metagenome before we move to machine intelligence and how classification really happens in metagenome. Okay, so we had finished with the assembly of metagenome and we had contigs, and so far what we have done, as is shown in this slide, is the 16S. Is the screen changing for you guys? Is the screen changing? Hold on 1 second. Let me. For some reason, my browser got frozen. So, okay, now it's fine. Okay. So, okay. So it's fine. So for some reason my screen was frozen. So we have done 16 sRNA, and how we annotate them. I think in the lab practical class, you guys must have done the 16S RNA analysis, and we also talked about why we need elite protein markers, because 16S RNA based classification can be rough because many very closely related species may not be able to differentiate using 16S RNA because they are very, very conserved through evolution. So we have this elite protein biomarker. We did that on the other day. We did a little bit of CRISPR-Cas. So what we're going to do is to go a little bit details on them and see antimicrobial. I will give you the databases, phage, plasmid, Rubisco, and everything, how we go about annotating all these in metagenome. Once we're done with the annotation, we'll go into the classification method. Okay. We talked about this on the other day. I'm just repeating this again. Because 16S RNA is a very crude measurement because it's very homologous between closely related species. Clear differentiation between the class taxonomy, you don't get that. Okay. So there are 40 protein biomarkers that have been created for bacteria and archaea. The condition for those 40 proteins are they should be there in all taxa. It's not like couple of bacteria won't have this protein, then you cannot do the taxonomic analysis using that. And they should be able to produce phylogenetic tree, meaning that they should be diverse enough to give you nice phylogenetic tree, but it shouldn't vary too much in terms of copy number. Same microbe shouldn't have 10 or 15 copies of the same thing. Okay, so that won't work. So they picked up these 14 protein biomarker. We have FASTA sequences of that, which you may be able to take this weekend if you've already this week practical class. If you have if you're already done with the 16S rRNA thing. I have asked Afia to take all the dendrogram, that phylogenetic tree that you may have generated for 16S RNA, and send it to me. So I want to know how many people were actually able to go through the process and give me that, produce the phylogenetic tree. So we will do the protein-based biomarker. The reason to do the protein-based biomarker is it's going to be slightly different. And when you do this in the practical class, first difference is you're going to have to do not blast N, because it won't be at the nucleotide level. Now you have a protein sequence, because protein biomarkers are protein markers are protein sequences, and you're going to have to find protein level homology with the contigs. Contigs are the nucleotide database. Contigs are nucleotide. Your query sequence will be protein. So you're going to have to use T blast N. I think it is T blast N you have to use, because the database is nucleotide. So it's going to translate the nucleotide into all 6 reading frames and try to find the homology to the query protein. Okay, that's what it will be, which is opposite for what you have done with the 16S. In 16S RNA sequencing, what you did was you took the contig as a query sequence and you blasted it against the 16S RNA. So your query sequence was contig and 16S RNA accession was the database. Here it will be just opposite. Your protein markers will be the query sequence and your contigs will be the database. So in any event, you guys have learned. I hope it is not for first time you have done that, but if it is the first time, I am happy you guys did that, to create a tabular output for BLAST. Tabular output for BLAST is beautiful because it gives you the query. First column will be query, second column will be database hit, third, fourth, percentage homology, gap, start, end, and e-value. And you can also have your own customized columns. BLAST will allow you to produce customized columns. Sometimes if you don't like the default column identifier, you can create just a customized column. In any event, and there are some formats like out format 6, out format 7, out format 8. Those are already default column settings. So out format 6 is what we are using in our 16S RNA blast output. Mostly I prefer using that, because then it's always standard in your mind. You always know which column is what. You can't keep on changing the output columns and every time get confused. I don't know which column is what. Okay, so use out format 6. That's what we used for 16S RNA. I think that's what you were told and that's what you used, right? Okay, so now we'll use exactly the same thing, but here it will be protein sequence. And here you're not expecting for 100% homology or anything. So the e-value cutoff, you have to be carefully picking that. You got to look at the alignment sometimes and then come back and pick up the correct e-value and percent homology from the blast output. So I definitely want you to do it at the protein level as well, because blast is the most confusing tool molecular biologists got. It's easy to use, but it's very hard to interpret. So I want you guys to do this in the next practical class. So what happens is this PubMed ID, I already told you last time, this PubMed ID is where they found this 40 things. So you could now use 16S rRNA and these 40 proteins to create a phylogenetic tree. And why are you doing the phylogenetic tree? You want to know the taxonomic composition of soil, or a gut microbiome, or skin microbiome, or saliva microbiome. You want to know the taxonomic classification of that. That's why you're using all this. Next thing, what you want, why you're using it, you're comparing two samples. You're saying from this soil and that soil. Here in this soil, nothing is growing. In this soil, everything is growing. What is the difference? What is the microbial composition that is different that makes this soil very rich and this soil very poor to grow something? Okay, so that's a problem we're having. So to do that, that's what these taxonomies are good. You can say that okay, this particular family is not present in this particular phyla or family or genus is not present in this soil and it's present here, and so on and so forth. So you can do literally differential taxonomic classification using these two tools. Other things what we can do. Let's see. This is one of the similar kind of dendrogram you will get this week in the practical class when you use the first protein marker against the four data set. So you have a data set, use the protein marker to produce a tree like this and compare that with that of 16S RNA to see whether there is something that changed. Is the resolution a little better with the protein biomarker or not? Okay. Okay. The other annotations, what you can do, what people really, really want to find out is antimicrobial genes. Antimicrobial genes, sometimes you want to discover novel antimicrobial genes in metagenome samples because there is too many novel microbes that will be there. So there is a scope to discover them. So antimicrobial genes, again, you need a database for that, right? For 16S rRNA sequencing, you had the green gene database. For antimicrobial genes, you have a database called CardDB or MegaRS. Okay, I think RES, it's Mega RES actually. CardDB and Mega RES. And we have these things available in IBIB servers, and some of the senior students will allow you to download that for this Friday practical class. And I also have the public link. Later in this slide, I will show you public link for these two databases where I got them. Okay, plasmids. Then there will be a lot of plasmid in the contigs. How did you get the contig again? Just to recall what we did. We just took the entire soil sample and we extracted the DNA and we sequenced it. So every microbe must have a plasmid, and the plasmid DNA also went in. So some of the contigs must be plasmid. Okay, so we need to find out which contigs are plasmid. The reason to find where the plasmids are, because that's where sometimes my antimicrobial gene will be sitting there, because they get shuttled between microbes via plasmid. Okay, so how many plasmids are there? Are there new kind of plasmid that is sitting there in some novel microbes? Okay, that's so plasmid database also we need to download and see. And then you have phage. Because microbes get infected by phage, there may be some microbes that are infected, so phage must be living along with microbes. So can we detect the contigs? So you got the DNA for the entire thing. So if the phage is there, you would have phage contig sitting there. Plasmid is there, plasmid contig will be there. So everything will be in one of the contigs. So you have to identify phage. How do you identify phage? You got to think of a way to identify phages, because there is no DNA. So it's not like 16S rRNA where somebody has created a database of all the phages and stuff. So some of the phage HMM models are sitting there. So I created a phage specific HMM set of HMM model for finding out this. You may be able to create more. So this is not, it's not end all. So you can look into the HMM databases and find out your own phage specific model to see in your contigs which phage protein is sitting there, which contig has a phage protein in it. Then you have the CRISPR spacer. We did talk about this on the other day, and we'll talk a little bit more today because that's very, very interesting. Then the other thing you could find is an integrative conjugative element. Integrative conjugative element is it's a stretch of large number of proteins that has a definitive order, which whole thing gets integrated into the from microbe to microbe. They go from plasmid, they go into integrated into microbe. So there is a linear stretch of protein sequences that's known. It's called ICE elements. So people want to find out that to see whether which kind of protein got shuttled between two microbes. Okay, so that's one thing. And then the metabolic pathways such as energy. I missed a G there, but energy or carbon metabolism. Okay, which soil has what kind of a metabolic genes active at a given time. And then you can also look for COGS, cluster of orthologous genes, to classify. So all these things you can do. I'm pretty sure much of this is also mentioned in your microbial classes. Like Shivkumar must have taught you all this, so you're familiar with that. But all these things you can now bioinformatically search in a sample, soil or gut or something else, where multiple microbes are sitting. So you can analyze them. So where do we get all these databases? Yeah, there are too many questions people are asking. How do I find out? So the first way to do that is, what are all the things you want to find out in the sample? Sometimes investigators will know what they want to see. As a bioinformatician, sometimes I'm going to mention something here. And you don't want to just do what is asked of you. Okay, you can also get more creative. You can say, oh, I can find out this because someone else has found out that. You can do some research. So either you can act like a technical assistant, just whatever is. Somebody asked CRISPR, that's why I'm doing CRISPR. Somebody asked me this, that's why I'm doing this. Instead of doing that, you can be motivated and you can say that, okay, I want to find all this. There are too many other things you can do with the metagenome. Okay, these are certain things I have I have tried to understand and work, because some of my collaborators have asked for these things. Okay, that's all. So when they ask for these things, then how do I do that? How do I find out phage? So you have to do some research on it. Okay, and there may be better ways to do that. So you can think of how to do it. Okay. So for example, okay, this is a slide I'm putting here. Okay, in this slide, what's happening is, when you talk about annotation, you're talking about um, CRISPR, CRISPR spacer, right? So when you're talking about annotation, you have CRISPR, spacer, 16S RNA, plasmid. These are all blast. Has the screen changed for you guys? Has the screen changed to annotation title? Okay, so I don't know why my screen is not changing here. Okay, that's fine. Thank you. Thank you. So blast 10. So some of them you have to use blast 10. Some of them you have to do the DNA to DNA search, because CRISPR and spacer, if you translate it, it will give you nothing. Okay, that is meaningless, because its function is at the DNA level. 16 is RNA. The function is at the DNA level, RNA level. So there is no translation. So you do blast, you do find the homology at the DNA level. Plasmid again. Plasmid, I guess there is nothing like a conserved protein or something in the plasmid. There is no recognizable element in plasmid. So you can take all the known plasmid DNA and you just blast the contig against them using blast 10. So it's important to know which blast to run. Okay, at the same time, when you're doing protein level homology, like finding antimicrobial gene, finding metabolic pathway, genes of the metabolic pathway, finding a cluster of orthologous genes, you want to blast at the protein level, meaning that the alignment or the homology that you're finding has to be at the protein level. The database can be a DNA database. For example, your contigs are DNA. So you can use them as a DNA database to search for antimicrobial proteins. So this particular slide I have told you blast and what when do you use blast and when you use the protein level T blast and then when you use HMM. Why am I going to HMM now? Why can't I keep on doing blast? Okay. I I hope you guys learned HMM and blast and everything very well. What's the why is how is HMM different? Suppose you don't have a single protein. You don't have a single protein and you want to search a database for a class of proteins. A class of proteins means like all kinases, for example. So you don't want to do. Suppose I have phage protein, and that varies from phage to phage. It varies. So how many protein sequences am I going to search the database with? Like there are 100 phage proteins, same protein from 100 different phages. I'm getting the same protein. They have diverged. So every protein is a different protein, but they all function the same in different phages. So I have to do 100 blasts with my database to make sure that I didn't miss anything. Okay, because you can use this the protein from this phage. You didn't get anything. That doesn't mean that there is nothing there. Maybe the other phage may have a homology there. The third phage may have a homology there. Okay, so you have to blast with everything. What HMM does is it has taken a given protein from many diverse phages, okay, and it has created a model for you. So you do one HMM run and you've covered it all. You're getting it. Why you have to use HMM here? Why not blast? So what you could do to find phage is see a phage specific protein for which a HMM model is available. And then you could take that model and search the contig database using that model. Okay, this is also something I want you guys to run. I don't know whether you guys run HMM and found something in your second semester class, but I do want you guys to take one or two HMM model which I have found and I will give it to you. So this Friday practical class, you will take CardDB database, you will take the protein marker, you make a dendrogram, CardDB, blast CardDB against the contigs and tell me which contigs have decent homology hit to antimicrobial gene. And third experiment you will do is to take a HMM model of a phage and blast it, search your contig database to see whether there is any contig that you think belongs to phage. Okay, these are three experiments you're going to do. So that's a practical for this Friday. Okay. So here, these slides maybe I should send to you guys so you guys can keep it with you. 16 is RNA where in the public domain these databases are available. CRISPR database is available. Next one, spacer database has been downloaded from this side, and plasmid database has been downloaded. These are all the DNA database you need for running BLAST10. Alright? And then at the protein level, you got the phage. There's a phage database that's sitting there. Although I did the HMM, there's a phage database that's sitting there. So I give you the link here. MegaRes, that's antimicrobial thing. CardDB is again an antimicrobial database. So these are all the protein database. Now you've collected all the databases. There may be more needed, but I'm saying for now, for a preliminary annotation of a metagenome assembly, you got most of this thing collected now. Okay, for phage HMM, All these HMMs I've collected. Okay, so there is a phage lysis gene, phage fiber gene, phage TTP, phage lysozyme, phage integrase, phage DNA binding. These are phage specific HMM model. Okay, so we have collected all that. So I will send you a couple of them so that you can run and you can find out. So these are also a good practice run for HMM, blast, and other things. You've learned a lot of theory and a lot of other things, but you never had a chance to practice it. For practical purpose, like practical use, what is the use for these HMM and blast, right? So this is one class you will learn how to what's the practical use of HMM and blast in metagenome analysis. Okay. Then there is CRISPR database. As I told you, I downloaded CRISPR database. So is this slide really clear to you guys? Are you guys able to read numbers clearly? Yes? No? Uh-oh. Oh my God. Okay. Okay. Let me see whether I can blow it a little bit. Now? Is that better? Okay. Okay. So I'm going to have to scroll back and forth a little bit, okay? Because I just magnified it. So here there is a blast output. Here there is a blast output, okay, where um, the first from here to here, I don't have a pen today, as I told you, I have a crisis with my pen situation. So, do you see all the way the first, there is a gap here, first all the way up to underscore 3? That is the contig. That's a contig, okay? All the way coverage underscore whatever underscore 3. There's a space there. And then you have a NC underscore all the way to 23, right? So the first part is the contig, which is a perisequence, and the second part is the accession for the CRISPR database. Okay, NC underscore all the way going into underscore 23 is a CRISPR database. Okay, that's a CRISPR accession. Okay, then I'm going to have to come back here. Hold on. Okay, okay, and then you have 100%. That means the contig has 100% match to this CRISPR in the database. Correct? Okay, and then it says this is again. Oops, sorry. I don't know why it's not going. Hold on. Hmm. Hold on 1 second, okay? Okay, it got. Hmm. Okay, anyway, it won't move anymore. Okay, hold on. Okay, this is. I wanted to show you something that. Okay, now it's. Hmm. You're not seeing it. Okay, now you're seeing it. Okay, good. Okay, so what's happening here is, so I told you the first column was your contig, second column is CRISPR hit, third column is 100% hit. The fourth column is, what is the size of the CRISPR? It is 37 bases. Okay, this is where the beauty is. Forget the next two columns. I think it's zero gap, means a full 100% identity, and zero gap. And the next zero, I'm not sure what the column is, but look at the last two columns here. Do you see the numbers here? Okay. The beauty of that is, go back to the bottom here where I have my pen, this one. Do you see that where my cursor is? Okay. 47290. This CRISPR is starting at 47290 to 47326. Okay. Then next is 47326. It's ending at 47326. And the next one above that is starting at 47365. 37 bases away again. Do you see the gap? So this CRISPR element is hitting from 290 to 326. The next CRISPR in the same CRISPR, the same thing in your contig, it is hitting to the same CRISPR sequence from 365 to 401. Okay, and then 439 to 475. Then it's going 512 to 548. Then it's going 586 to. If you look at each CRISPR, it is separated by 30 or some space, and the next CRISPR is hitting. Then there is a 30 or 36 space, next CRISPR. It's there in tandem. Are you getting it? Yeah? So, meaning that in the blast you will see that there are that many CRISPRs repeating. So, in this particular case, how do I know it's repeating? Because the CRISPR accession is exactly the same. So, your contig is exactly same. In the same contig, it's picking the same CRISPR element at different regions, 37 bases apart. So how many CRISPR sequences are there on this contig? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. There are 10 CRISPR sequences in tandem, separated by spaces. And the spacer, exactly you know how much a space is, because 326 to 365 is the space. And the next CRISPR starts in 365. First starts at 290, next starts at 365, next starts at 439, 512, 576, and keeps on going. Beautiful. Okay. Unless you've done this blast, you'll never understand CRISPR spacer so beautifully. Right? So again, in the same slide, this is another contig, not the same contig. So this is one contig that has CRISPR. There's another contig that has CRISPR. So in this, the space is so less you can't see the two columns separated. Actually, up to _1 is a contig ID, your query sequence. Okay. After that, it's NC underscore all the way to 32. That is a CRISPR ID. This CRISPR ID is different than this CRISPR ID. Okay. So this is a different CRISPR sequence, and in a different contig. It again has 100% identity. It again has 37. Okay, and the same thing. It starts at different position and goes. It's actually in different position here. You can see, right? It's at 39453 to 489, and then 528 to 564, then 602 to, and it keeps on going. Here also you got kind of 9 CRISPR element in a row. So you find large number of contigs that has CRISPR in tandem. Okay. As I explained to you on that day, so there is a good good reason for you to think that you can discover novel CRISPR element. You can discover novel spacer elements. So again, to explain to you what the CRISPR and spacers are, CRISPR is a property of microbe. Microbes uses this CRISPR repeat to act almost like an immune system. Immune T cell, T memory cells, right? So every time a phage infects it, it will take a small snippet of the phage genome and incorporate it as a spacer between two CRISPR elements. Okay, that's how it's going to remember that next time when that phage comes, it's going to chop it off using the Cas9 protein. Okay, so the more a microbe gets infected, the more the CRISPR will keep on, because each spacer is going to be different. All the CRISPR element will be exactly same. The spacers will be different, coming from different phages. So more a microbe gets infected, more CRISPR elements will be there and more spacer elements will be there. So obviously the spacer elements between these are not going to be exactly conserved like this. Okay. So I'm going to go shrink this a little bit and adjust my screen. Sorry, my pen is not working today. Hopefully by tomorrow I will have that working. Okay. So now what do you expect from spacer? Right? As I said, CRISPR is a property of a microbial genome, so it is conserved. It will have same CRISPR repeating like that. Spacer won't be repeating like this. Spacer is coming from phage. Okay, so the spacer database now has all the known spacers. There is no similarity between spacers, but spacer is recognized because it is between two CRISPR elements. So they have created a spacer database. I gave you in that previous slide, I gave you where the spacer database was downloaded. Okay, so now if you look at the spacer, element, of course, it's again very small. Without even going through the detail of this, first column again is the query, your contig. Second column is the spacer accession. Okay. If I blow this a little bit, make it a little bigger for you guys. These are much smaller numbers, so it will be easier for it. So again, here this is your spacer accession, and because I moved the screen, my query accession went away. Okay, it's 100%. Of course, spacer size is slightly varying, but same size as CRISPR. Okay, about 38, 37, 36, 35. Okay, so two CRISPRs will be separated by a spacer of the same size nearly. Okay, so here if you look at it, is it in tandem? Can you tell if it is in tandem? How will you know it is in tandem? These are all note that in the CRISPR element they were all hitting the same CRISPR element. Here they are hitting separate different spacer elements here because spacers are not conserved. Okay, they are hitting different spacer, but in your query sequence from 76 to 112 there is a spacer. Okay, and the next spacer is 150 to 185. So between 112 and 150 there should be a CRISPR. Okay, 150 to 185 is the spacer. 185 to 223 is a CRISPR element. Then 223 to 258 is a spacer. 258 to 296 is a CRISPR. Then you have 296 to 333 is a spacer. 333, and then there is a big spacer, there is a big gap. So from here to here, the continuous 5 spacers are nicely hitting. Okay, so now using your blast output, you can merge the CRISPR and spacer and see whether the CRISPR and spacers are interlacing like this in a same contig. Okay, that's the kind of homework you have to do. I'm hoping you guys are getting it. Okay, so it's fairly straightforward. It's just that, hold on. Yeah. So I'm back to the size. Yeah. Okay. This is good. Okay. So now you, because you ran the CRISPR database is separate and the spacer database is separate. How do you know that the spacer database and the CRISPR database are interlacing? Meaning that in a particular contig, the spaces within the CRISPRs are hitting the same contig. Okay, so you got to interlace that and you got to say that, okay, aha, I found. So in the next table, which I think I showed you on the other day, I again here you're going to say you didn't see, you're not able to see these things. I think on your screen also you could blow them a little bigger. Okay, so I'm going to move the screen slowly. Again, you got the contig ID. Then is CRISPR, is it there or not? I'm saying yes. This is my kind of a consolidating my results here. Okay. So see in the first, now on that day I showed this table. Probably it wasn't that clear to you. Now for this contig I'm finding 15 CRISPRs. Okay, I'm finding 15 CRISPRs, but I'm not finding that many spacers because, as I told you, spacers are not conserved. Okay, if it's there in the spacer database, it's there. CRISPRs are conserved. Now, what does it mean? Meaning that you have 15 CRISPR elements. Okay, only 8 spacers you're finding. That means 7 more spacers are novel in this metagenome. So you may be able to discover 7 more spacer element and add to a database. Those are new. There may be new phage that we haven't yet known about it. It may be coming from some phage that's infecting some microbe that we never know. Okay, so if there are 15 CRISPR element, they have to be separated by spacer element. Only 8 spacer is in the database. So the other spaces you can discover it. They are novel spaces. Okay. But here in the third line again, you have 10 CRISPRs and 10 spacers. So that's correct. Maybe that's a known one. Already known CRISPR genome. Already known genome. That's why it's eating everything. Again, in the fourth row, you see that there are 11 CRISPR elements. Okay. And only 4 spacers are hitting the spacer database. What does it mean? It means that there are 7 more spacers. You have just discovered it. Right, by running blast here. You've discovered 7 CRISPRs. 7 spacers, novel spacers. Again, so anytime you get CRISPR and you're not hitting spacer, that means you're discovering novel spacers. Okay, that's the beauty of it. Okay, so I think once you do it yourself in the class, you will get a better feeling for this. Okay. Okay. So for the plasmid also I did that. Sometime you have to compare 4 different samples, soil samples. Here I'm showing 4 different soil samples. I ran it against the plasmid database, and what I'm finding is large number of plasmids are common to all 4 samples. Okay, 79 of them. There are many unique plasmid in one sample, like 193 in AW5. That's the name of the sample at that time. Not that many unique for AH1 and WM and AK. So, what it means, probably a microbiologist would know better. To me, I would interpret that this AW5 is a very unique sample, which has very many novel microbial species with unknown plasmids, and that's why they are so unique here. Okay, that's my interpretation. But anyway, people, well, it also depends on where you got the samples, what you're looking for, and what's your question. You can interpret these results. So this kind of Venn diagram is also very useful. So now, what is our challenge here? Okay, we are almost coming to end of the lecture, like maybe 15 more minutes. But annotation. So annotation. So you get annotation as text files, right? Like when you run a blast output, you got a tabular output. You got 16S RNA tabular output. You got plasmids tabular output. You got phage tabular output. You got, so all these outputs as, you know, different, sitting in different places. How are you going to consolidate the results, right? You you have known, your known genes, unknown genes, novel genes, antimicrobial genes in this context. You have metabolic genes and pathways in this. In this, you have to consolidate all these scattered output sitting there, and you have to somehow summarize that and say that what does it really mean in your sample. Okay, so basically, one thing is consolidating for a given sample what's going on. Second is, how do you even compare between two samples you're comparing? 16 is RNA, we did that already. In the class, you're going to say, I'm going to create a phylogenetic tree for sample 1, sample 2, sample 3. I know which um, taxonomy is present and absent and that kind of thing. And sometimes the abundance of the taxonomy also can be different. Even though both of them have the same taxonomic structure, the abundance may vary between, between sample to sample. Challenges in comparative analysis across samples. Okay. Okay. So this I'm showing here one kind of results how you consolidate. On the left you're seeing, right? Okay. Now it's a little bigger. You see that there are five or six samples here. I taken it from a published paper. How do you consolidate results? As a bioinformatician, that's a major challenge in metagenome. How do you consolidate results? Because when you send those text files to the investigator who has asked you to analyze it, they can't even handle these text outputs. They don't know how to consolidate the result. They want to see something like this. So you can generate this kind of software in R, okay? But you need to be able to parse the results from all the text files and put it in a visual format like this. For example, here in the left, this colorful heat map, it tells you which microbes are more abundant in which samples, almost like a heat map of the presence of a microbe. Red means it's a lot present and gray means it's not present and stuff like that. So you get a differential microbial abundance. On the right, in the black and white, what it says is whether, uh, you know, electron donors. Electron donors, all the metabolic pathways that are involved in electron donors, are they present? Electron acceptors, are they present? So these are all different metabolic pathways if it is present in a sample or not. Okay, that's a kind of annotation here. This kind of representation is beautiful. On the right here, LAU4, LAU10, they also are unique to some metabolic pathways are known to be in a particular phylum, particular family, particular genus. So based on that, you can tell that this belongs to that classification. Okay, so lot, lot of work you have to do after that to consolidate your results, and this is still a huge challenge. Okay, you think that, okay, annotation, okay, I know how to do blast, I know how to do HMM, I know how T blast 10, I got everything. But what are you going to do? These are all big text files sitting there. You cannot, if you cannot summarize it nicely in a biologically meaningful way, all this analysis is no good. Correct? So that's one thing. On the right, yeah, on the right, what you see here is called. This is how it looks like. These are all the clusters. This entire sample, we're going to learn how to do this. These are called self-assembling map of all your contigs, or how they are separated. They are forming nice clusters. Each cluster may be a particular um, very closely related species. Each cluster nicely gets separated. Okay, this kind of, so on this kind of thing they have classified. I hope I can move it. Let me see. No, I can't move it. Okay, sorry. Anyway, okay. So there's annotation on all these, which cluster belongs to which metabolic pathway. So they've annotated all that. So it's still not possible to do these things by hand. So we have to have methods, and we have to know enough biology to be able to consolidate our results and find and report our findings. Okay, that's the most important thing. Okay, okay. So what have we done so far? Annotation. Annotation is always the most difficult thing in genome analysis, not just in metagenome. Even when you have a regular plant genome sequencing done, annotation is the biggest challenge. How do you automate it? How do you consolidate it? How do you report your results? How do you make sense out of all this annotation that's coming out of it? That's the biggest challenge. Okay. One of the other challenges in metagenome is the coverage is unknown. When you have done coverage, tomorrow in the quiz I will ask you again to see whether you are able to understand what coverage is. But coverage is usually what? If I have a genome of 1 million bases, let's say, if I want 100x coverage, then I will probably ask somebody to sequence 100 million base pairs. That way I'll get 100x coverage. 1 million base pair is 1x coverage, 100 million base pair will be 100x coverage. But in metagenome, in a given sample, I don't know the genome size, because the genome size of metagenome is equal to how many unique microbes are sitting there, and what is the size of their genomes. We don't know that. We don't know how many unique microbes are there. We don't know the size of those genomes. Okay, so we have no idea. So we don't know the coverage. So you just arbitrarily sequence some amount, and you are assembling it. Your assembly is as good as the coverage you've given. If the coverage is low, you won't be able to get good assembly. But we don't even know what coverage is. That's a major problem with us. Okay. And then the other problem with metagenome is not only that there are unique microbes, okay, each microbe will have different abundance. One microbial species can have huge number of copies in the sample. Others may have only very few copies of that microbe. So that variation also need to be accommodated for when you're looking for sequencing, when you're doing coverage. Okay. So, and then the other thing someone mentioned on that day, there will be too many species that are very closely related. How are we going to deal with that? Because of that, we'll get sometimes the contigs will get merged into two different species, but they're very closely related. Assembly programs cannot tell them apart, so it will put them together. So you will have chimeric contigs. But if the abundance of those species are different, then you should be able to find out based on the abundance. You will say that this whole contig is there. The first part of contig, you got too many copies of reads coming from first half of the contig. The second half of the contig, you only have 2 or 3 copies. So that means this is the chimera. So you can split them nicely. So those are secondary screening you have to do to remove the chimeric chimeric thing. So these are all the challenges with metagenome. Because this is a challenge and you have already spent so much money sequencing so much, you still didn't get good data because you didn't know the coverage. The sample was too complicated. Because of that, now we're going the reverse order. Actually, we're now going to do a simpler one. Because of that, now people use first step in a metagenome sample. If you know nothing about the sample, okay, first thing you do is not the metagenome whole genome sequencing. You don't do the whole genome sequencing. Okay, what you do is you only amplify a particular region from the 16S RNA, 16S RNA, and that amplicon. Okay, so because 16S RNA is present, okay, what is a chimeric contig? Chimeric contig, okay, when you're assembling, what you're doing? I'll answer that question before I move to the 16S sync RNA. I'm going to go to the previous slide. Okay, so our question is what is chimeric contig? Okay, so suppose you have two microbes. They're unique. They're unique species. They're different species, but they're very closely related. Their DNA sequences are not that different. So what happens is, remember when I taught you assembly, okay? What happens? You have to find an overlap, okay? You're finding an overlap of 31 mer, let's say. So there is a read here, there's a read here. But if it so happens that that 31 mer between the two species are very similar, okay, you're going to start because although this read is coming from a different species, because the 31 mer overlap is the same, it's going to start running into different species now using the overlap. Okay, so the first part will be coming from one species, and suddenly because the overlap is very similar, because the species are similar, okay, it's going to start running into the different species. So that's called chimeric contig. So this half of the contig is representing one species, and this half of the contig is representing very closely related species. Okay, that's what I'm meaning chimera. And the reason why I'm saying that you can separate them, if this species is say 100,000 copies of this species is there in the sample, only 100 copies of this species is there. When you have a contig, this half will be very highly covered. This half won't be covered at all. So from that you can tell it's a chimeric contig. So we can clean it up later. You're getting it. So there are a lot of difficult procedure people use to clean up the contigs finally, so that you can get unique microbes in unique contigs. I hope you got it. Okay. So this was a long run. All the annotation was so complicated. Genome assembly was complicated. It was big, expensive process. Both sequencing was expensive and analysis is expensive. So what people do is a much simpler thing. Okay, this analysis of this is very different. The pipeline is very different. Not this Friday, but the Friday after that, you will do 16S RNA sequencing analysis. Here, what they do is forget about genome sequencing. All you're going to take is a soil sample. You take the soil sample, extract the DNA, okay, but don't sequence the entire DNA. Extract the DNA and amplify only a particular region of the 16S rRNA, which is V1 to V9. There are 9 regions you could amplify separately. So V3, let's say V3 is this yellow box you see here. This yellow box is a V3 region. So there are two conserved region and a varying region in between. Okay, it's conserved and in between it's varying. So the conserved region is conserved across all microbes. So you made a primer out of the conserved region. So when you make a forward primer and reverse primer of the conserved region and you amplify all the pieces from the entire metagenome, every species 16S V3 region of the 16S RNA from every species in the sample will get amplified. Okay, you're only going to sequence that amplified fragments of 150 bases. I think V3 is 150 bases. So you already get 150 base pair fragment amplified and you're going to sequence it. So you're not sequencing anything else. It's all V3 region of the 16S RNA from every species that was present in the metagenome. And now the problem gets simplified. Okay. Now it's cheaper because now you can do a lot. You can very small amount of sequence, you can get large number of species can be represented. Okay, I told you when you have a targeted sequencing, okay, it's only 150 base now. It is not 4 million. The genome size is 4 million for microbe. Where is 4 million? Where is 150 bases? So with a very small MySeq or something, you can really sequence very deep and you can classify. 16S classification that you did with 16S RNA, here it's slightly different. It's not the same way for 16S RNA sequencing. Metagenome sequencing, you did that last Friday. Here, this again is very popular method now. I will tell you that there is a gut metagenome project in NCBI database, gut metagenome project. Tens of thousands of gut metagenome has been sequenced using 16S RNA sequencing. Okay, where all they're asking is from diabetic gut to a normal gut, from cancer gut to, so they want to find out how the gut metagenome changes across population. Okay, for that they're only using 16S RNA sequencing because that in itself gives you good picture of what is the composition of a metagenome. Okay, so this is simpler method. Tomorrow's lecture will be covering how to analyze 16S RNA sequencing, because in this sequencing you're not going to do antimicrobial genome, CRISPR-Cas, there is nothing there. This will only have the V3 region of the 16S RNA. But it's a short segment, so the analysis is going to be slightly tricky but very clean. Okay, so we will do that tomorrow. If possible, we will do quiz. I'm hoping all of you can get the stream chat working by tomorrow, so that we can all. I will try the quiz anyway. If some of you are not able to use the stream chat, then I guess you won't be able to participate tomorrow in the quiz. Okay, that's how it's going to be, because you will only see. You may be able to participate. It won't score you though. Okay, so as long as you can see the quiz board, you can answer it, but it won't be able to score you. Okay, so that's a problem there. Okay, so I think I'm going to adjourn at this point, so at least today you can go to your next class in time. Okay, so see you tomorrow for quiz. Make sure that you find out the right kind of browser, and also ask your friends, whoever is able to get to stream chat, make sure you have the same version of the browser, And also maybe you can upgrade your own operating system too. Sometimes operating systems are too old, but I think basically you just need to upgrade your browser. Okay, update your browser. Okay, thanks guys. Bye now.